And on behalf of the IMarEST, a very warm welcome to Trinity House for the latest in the Stanley Gray lecture series. In order to introduce our speaker this evening, I'd like to pass you over to our Senior Technical Advisor, Dr. Philomène Verlan. Philomène.
Welcome. Thank you so much for coming tonight. You're in for a real treat. I'm very, very personally honored and professionally privileged to be able to introduce to you our speaker, Dr. Cindy Lee Van Dover, who is a representative in one person of the three constituencies that we represent at IMarEST, the Institute of Marine Engineering, Science and Technology. Cindy is all three, does all three, and has been able to describe particularly well, not only in a scholarly way, but also for the rest of us who are out there interested in the oceans, exactly what it feels like to be physically present in it. When you came in, the music that was playing is the music from the song The Octopus's Garden, which is, I think, very appropriate. And this is also the title of her book, which I recommend to you all, particularly the students among us, because it describes very well the varied routes that Cindy took to be where she is today. There are many different paths that you can take to work in the oceans, and this is one example of how you can achieve this type of professional career. There are others out there. The Institute of Marine Engineering, Science and Technology has a careers program that is specifically dedicated to making these possibilities available to as many young people as possible. And this is quite important because the ocean covers, as you know, 70% of our planet. We should probably be better called planet ocean than planet Earth. And the variety of activities that we can engage in the ocean, both professionally and personally on a leisurely basis, are so varied that there are infinite opportunities for employment and for enjoyment. Now, to come to this evening's lecture, you will be receiving an insight into deep sea exploration, because most of the ocean is, in fact, deeper than 200 meters. It can go down as deep as you know to 11,000 meters in the Marianas Trench. And Cindy has been on top of it, in it, under it, and she will now give you some insights in the next 45 minutes or so on what it has been like, what her reflections are, putting it in a context of our three constituencies again, the engineers, the science, and the technologists. And at the end, we will be opening the floor for questions. Thank you very much. Welcome, Cindy.
Thank you. Thanks very much. It's a real honor to be here. I really appreciate the hospitality that I've been shown so far. Dr. Verlan, it's just been a great visit, and it's just a thrill to have a chance to talk to you about something I love so much. I took a little bit of liberty over what I was asked to talk about. I want to talk a little bit about deep ocean industrialization as well as exploration and responsible stewardship, and in particular extraction of ore from the deep sea. And I'm going to take you there by a little bit of an indirect route. So I'm a professor and I give outlines. Sorry, this is just the way I work. So I wanted to make sure you kind of had a roadmap to where we're going. So I'm going to tell you a little bit about how we access the deep sea. You've probably seen some documentary videos and got a sense of the submersibles and other tools that we use these days, but have some new vehicles that we've been using that are really very exciting. I'll give you a little bit of biology or geology of hydrothermal vents, the hot springs on the seafloor, so we're all on the same page about these sites that are hosts to seafloor massive sulfides, to ore bodies of commercial interest. We'll talk a little bit about mining interests and potential impacts on the seafloor. And then I have been working with a mining company and an academic industry partnership, and we're thinking about how do we collect the baseline data needed, how do we think about mitigation of mining on the seafloor. So this decade. Well, I started thinking about deep sea mining in 2005, and I think this decade, the next, are really going to have the possibility of seeing the emergent deep sea mineral extraction, which is pretty extraordinary. Okay, well, so what do I mean by deep sea? For me, the deep sea is anything greater than 200 or 300 meters. That's kind of the technical definition. For me, it's really where there's no light and where there's no longer possibility of sunlight-driven photosynthesis. So we're talking quite deep, and I really work about 1,500 meters and deeper. 1,000 meters is shallow, and I'm not so interested in that. So I like to work 1,500, 2,000 to about 4,000 meters typically. So was James Cameron's dive to the Mariana Trench as big a deal here as it was in the US? That was the one day, at least in the US, where I think everybody thought about the deep sea for at least one day. And that's not an ordinary occurrence. I think other than that one day, I may be the only person who thinks about it year round. This is my baby. This is Alvin. This is a US submersible. It's a human-occupied vehicle. And it has a pilot and 2 scientists. Right after I finished my PhD degree, I decided the best way to be an ecologist of the deep sea was to get down into the seafloor environment frequently, and it's the pilots who do that. So I trained to become an Alvin pilot and piloted her. I was pilot in command of 48 dives. And I've done I think I've almost reached 100 dives total in my career. I would be happy to talk to anyone after this about what it's like to train as a pilot and be in the submarine, but that's not what I'm going to talk so much about today. One of the things that I have done, though, is in the past years related to this vehicle, is she's just been upgraded. She's just going through her sea trials and being recertified to dive. Everything's been rebuilt. Her entire hull, for the first time in, I don't know, several decades, we built a new personnel sphere out of titanium in the US, and it was a major, major undertaking. And I had the privilege of being the chair of the oversight committee for that. So every 2 weeks for about 4 years, I was on a teleconference for an hour or two, talking with the engineers, the project manager, to watch this thing come through. So this is her. This is after her construction. So she hasn't done a science dive yet, but I'll be taking her out in May as a scientist to use her in the Gulf of Mexico. So Alvin is untethered. So when you go down there, the pilot and the 2 people, you're not connected to the surface at all except by underwater telephones. You use acoustics to talk back to the surface. What's more commonly used these days now are the ROVs. This is our US ROV, scientific diving ROV called Jason 2. And here's the control van. So you take Jason 2 out on the ship. You take a control van, just shipping vans, fitted out with lots of screens and pilot controls and scientists' workspaces. And you work on the ship with this vehicle. And you don't have to dive, you don't have to be in the submarine. Or in fact, I should put it, you don't get to be in the submarine. You watch video instead of being on the seafloor. It's quite interesting. You'd think that somebody who trained to be an Alvin pilot would be just passionate about diving in Alvin. But in fact, I prefer, if I have a choice, I'll go out with Jason. And the reason is I've already been in that submarine. I can translate now the 2D into 3D because I've had that spatial experience in the submarine. And this is a much more democratic process. So everybody, the science party can all pile into this van and watch the science going on. We have repeater screens around the ship, so you can be anywhere on the ship usually and be able to see what's going on the seafloor. So the students, the scientists, the galley crew, the captain, everybody can be watching the discoveries made on the seafloor all at the same time. So it's a really exciting way to do science and much more engaging for the students on the ship. Although, if you ask my students in my lab if they'd rather go on an Alvin dive series or a Jason dive series, they all want to go with Alvin. Even though they might not get to dive, there's always that chance that they get to go to the seafloor. And so it's really very important and exciting the next generation to be able to get to the seafloor in person. It's kind of like going to outer space. So I wanted to show you a little bit of video from the seafloor. This is kind of apropos of nothing, but for you to see how these animals are collected. These shrimp are from the Cayman Rise. We were exploring there at 4,000 meter water depth with Jason. And we're going to sample some shrimp. And this is I picked a really good video clip, so it looks really, really easy, and they just come by the dozens. But that's most of the time, the pilots, when they're inexperienced, they have trouble, they drop the hose, so you really hope you have a good experienced pilot working with you. And then when the animals come up on deck, the scientists, we take them, and especially my team now is doing a lot of genetic work, so we chop up all the little animals. We look at them and take pictures of them, but then we parse them out into little vials and put them in all kinds of preservatives to bring back to the lab to analyze for the DNA. And we had an artist on board who really was interested. She was fascinated by watching a scientist process these animals. The vehicle came up at 6 o'clock at night, and we worked to about 3 in the morning. And she did a time-lapse picture, and I won't let it run the whole time. But this is kind of what it's like in there, and it was just constantly working on these samples. So when we say we're going on a research cruise, it really is a work job, not a kick back in the sun kind of experience. Okay, guys, you can stop working now. Okay. So I've told you about the human-occupied vehicles and then the tethered vehicles that are very capable. They have manipulators for sampling. There's another class of vehicle called an autonomous underwater vehicle. This one is must be being taken up out of the, whoops, I've lost my, it doesn't matter. It's being taken up out of the water, I believe. It normally is untethered. You drop it in the water, and it will just go on a program mission. So it'll sink down to the seafloor, and maybe if it's on a mission to do multi-beam bathymetry to look at the bottom topography, map the bottom topography, it will just do the program, mow the lawn, survey tracks that you tell it to do. And then it comes back up when it's told to, or when it's run out of battery power. So this is the US version, Sentry. All these vehicles are replicated, except for the human-occupied vehicles. There's not so many of them. Russia, China, Japan, and France have ones. But the ROVs and the AUVs, every country, a lot of the countries that have ocean interests are developing these. There's many of them around. It's not just US, but I'm from the US. Here's a really cool vehicle. This is one, it's called a hybrid ROV. And on the left, one is the mock-up, and the other is the lower frame is the real version. This is called IMarEST. It can operate in the AUV mode, which is what you see on the left, and so you go explore. So we like to go out and explore with the AUVs. They're very efficient at looking around the seafloor systematically. But then suppose you find the trilobites, right? You don't want to just have to go back to shore and raise a whole bunch of more money to get back out to get the trilobites, although that would be pretty easy if you found them. Instead, you want to have an ROV at the same time. So this thing turns into a remotely operated vehicle. And the way it does this is extraordinary. The engineers are so clever. They put it down on a 1,000-meter regular fiber optic cable with conducting wire. So that cable goes down to 1,000 meters. And then there's a junction, and it spools out an 8-pound test fiber optic cable. That's it. It's like a fishing line. It's like as thin as, well, I don't know how much hair. My hair used to be longer. Thin as a hair, pretty much. And it's 20 kilometers long, spooled up, and this thing can just run out 20 kilometers out. And they manage it so they're not tying knots in it. And it can go explore under ice. It can peek in under ice. It could go work in places that are a little bit more hazardous than others. And then when it's ready to come up, you break that line, you spool up the fiber optic cable. This thing comes up to the surface on its own. But all that time with that cable, it had communication, so you could sample with the manipulator, you could see what you were doing, have the video. So very, very interesting approach, and it worked incredibly successfully. Just this past summer, we took it out. And in fact, we use, James Cameron donated his experimental vehicle to Woods Hole, and we nabbed the camera off of that. So we were using his camera on IMarEST this summer. There's another way of working that's just breaking news. We're experimenting with how to do it really well as scientists. Bob Ballard's been doing it for a while. This is this telepresence concept. What you see here on the left is a shore-based laboratory called the Inner Space Center. And surely I can get this to work. I'm a scientist. There we go. So here's the shore-based laboratory. And you can see we have a quad screen. We can see 4 different video monitors. If we had an ROV on the seafloor, we could watch the ROV working. We could watch the eye in the sky vehicle that's watching the ROV working. We could look at the deck. We could turn it on to watch the sunset, different channels to watch different things. We took it out 2 summers ago now with the AUV. Now, what could be more boring than watching an AUV go in the water, because there's no signals coming up. There's no video signals. It's just collecting data on that survey. So we were experimenting with how to lead an AUV mission from shore. And so I was the chief scientist on this cruise. I still think of it as a cruise. I was in Rhode Island, and I was on a cruise, chief scientist. And we would plan the missions here in Rhode Island. We'd conference every day with the team on the ship. And they would give us feedback on what they were seeing, the weather conditions and ideas that they had. And then our engineers on this side would program the vehicle from shore and pop it over by satellite to the vehicle. So we were really doing telepresence in an extreme way. Incredibly successful. And we're not sure why it worked so well, but we got these students, there were 10 students there with us, who just sunk their teeth right into that data, came up with information about what we were seeing, plotting all the chemical information, looking at the photographs, telling us where to go next. It's going to be interesting. And by the end of the 10 days, they designed the mission, which is extraordinary. You normally don't, chief scientists don't normally let that go. That's the power position, right? But these kids were so good that we could just sit there and mentor them through it, but they designed the mission based on what they saw was interesting in the data. Really very powerful way to train students and to get science done. We had a blast. And then there's this, you can use the telepresence for live video feed and just the general public. So if you go on to the Nautilus Live website right now, they are diving at Kickhamjeni volcano in the Caribbean. You'll see, usually if they're in the water, you'll see the bottom video, live footage. It's maybe a second or two delay depending on the quality of your internet connection. They'll have the voiceover so you can listen to the scientists and the team talk. And when a big fish comes in the way, they're all going, "Whoa, look at that giant fish!" And so you can hear the excitement of what it's like. And then there's the boring times when they're over mud and they're just saying, "What's for dinner tonight?" So it's live, it's unedited, it's just how it is, and it can be really exciting. And then there's also and they also have a lot of education programs that go out to museums and classrooms. So it's a neat way to get students involved. They do professional programming every half hour. We were out with on Eric Schmidt's vessel, the Falcor, there's Nereus on the ship, and that's Eric Schmidt is the Google guy, and so he had everything archived. It was streaming live on YouTube, and it's all archived there, so you can watch all of our video. The only thing we censored was where we got tangled in a cable from the Japanese sub that was operating side by side with us, and they broke a cable and left it there, and we got caught in it, so we shut down the video at that point. But language. Okay, so that's a little glimpse of how we access the seafloor and some of the technologies we use. And what's exciting for me is because I was in Woods Hole as a graduate student, I know the engineers, and when they've got some new toy to play with, they often, I know about it, and I go to them, or they come to me and say, "Hey, we've got this opportunity, would you come play with us?" And so I get to see the vehicles when they're coming straight out of, when they're in test mode, which is, for me, really the best time to use them. It's more fun even than when they're kind of just standard operating procedure. I'm going to turn now and tell you a little bit about hydrothermal vents, because that's kind of what the rest of this talk is about, the hot springs on the seafloor. And my head may work a little differently from yours in that when I look out at the ocean, my mind goes to this image. I see it drained. And so I see the mountain ranges. This is the Mid-Atlantic Ridge, the East Pacific Rise, Indian Ocean, southwest, southeast, central Indian Ridge. Those are seafloor spreading centers where the plates are spreading apart. They're volcanic. So there's hot rock, and when you have hot rock, there's cracks and fissures in that rock. Seawater percolates down into that rock, chemically modified, comes back up buoyant with a really strange chemistry. So we've got the volcanic spreading center. This is off the Oregon coast to show you how this all works. And we get black smokers, hot springs on the seafloor. They're not unlike geysers. They're not intermittent like Old Faithful. They just keep pumping out until they until the heat source is gone or they get clogged. The temperature here, right at that orifice, typically about 350 degrees Celsius, so very hot. The smoke, they're called black smoke, or is that smoke is really finely divided mineral particles. So in fact, most of the ore body, most of the ore is going up in smoke, and it just goes into the water column, and it's distributed. It's actually what causes the formation, or contributes to the formation of manganese nodules and rare earth element deposits in the abyssal plain. And then there's these precipitated sulfides. And in some places, these grow up in 10 years. There's a volcanic eruption. They're plowed over with new lava and basalt, and they're gone. In other places, these chimneys last longer. They consolidate, they build up over time, and they form fossil ore deposits. When these things were discovered on the seafloor back in 1979, at that time, we didn't know that how fossil ore deposits like the Cyprus copper ores were formed. And this suddenly revealed how it happens. It's done on the seafloor, and the ophiolite of Cyprus is just uplifted onto land. So that's where many of the major ore bodies that have copper-rich ores come from, these hot springs on the seafloor. So instantly we thought about the commercial, economic geologists thought about the commercial value of these systems, but the technology really wasn't there to advance the mining or extraction of these. And also, along with this sulfide minerals, we find really strange animals that occur there, large abundance of animals. So there was in the US, there's a site off of Oregon called Gorda Ridge, and the US Minerals Management Service immediately had lease options, and were doing a draft environmental impact statement. And when they talked to the scientists, they learned fairly quickly that it's just crazy to try to do this now. We don't even know what's out there biologically and what we could be messing up. But we've come a long way since then. So here's a little bit of video to show you what a black smoker looks like. It looks like a smokestack. The fluid is, I told you it was hot, it's also acidic. So imagine if you were trying to mine around these areas where you have active venting, pretty caustic environment, really difficult, challenging place to work. So the technology that's going to be developed, or is being developed to work in these systems, has to cope with that. You've already seen a little bit of shrimp, so we'll skip that. Just a reminder, or wanted to let you know that just because I've been showing you shrimp, it's not always shrimp. You go around to different parts of the world's spreading centers and you find different types of animals. And in fact, part of my career has been exploring these mid-ocean ridges, chasing the biogeography, understanding the patterns of distributions of these species, which means I get to be a world traveler, and I got to go to lots of different places on the seafloor. I've been in every ocean. And we find that the communities are different, so we make discoveries. So colleagues in the UK, NOC, Southampton, discovered these strange hydrothermal vent communities dominated by crabs, just stacked on top of one another. Beautiful, beautiful animals. Here, this is from Lau Basin in the Western Pacific. This is also Lau Basin. These are barnacles, anemones. There's mussels here and shrimp. What supports this biomass is chemoautotrophic production. So bacteria are the base of the food web. It's no longer sunlight here. It's chemical energy that's allowing these communities to develop. And the animals are all exquisitely adapted to this very extreme environment, with especially very high levels of hydrogen sulfide coming out of the fluids, which is normally be toxic to you and me and to any animal, except for these guys who live in this special environment. They have biochemical adaptations to cope with it. They are dynamic systems. They're on volcanic spreading centers. Like I indicated, you can have eruptions on the seafloor. This is from the Western Pacific. And you can see when you're diving on the seafloor, these overruns of fresh, glassy, black basalt. And then this weathers away over time, so you get the older, you can recognize fresh from old. And we had in the 1990s, when I was a pilot, in fact, I dove at this time. There was an eruption in 1991. On the East Pacific Rise, so can you see this? This is Mexico, Baja California, East Pacific Rise Spreading Center. And there's a little area there where a geologist saw the bathymetry seemed to be inflated. And she said, I think there's going to be an eruption there. And so we went back, and she did that with remote vehicles. And we went back with Alvin, and I was with her on this cruise, and I was piloting, and we'd go down to the seafloor. There had been an eruption. And the dating, it was within a couple of weeks of when we got there with Alvin. Everything was glassy smoke, steam, it's not steam, but warm water was coming out of every kind of crack you could see. It was just amazing. It was just like this inferno. There were bacterial material blowing up, so that it was, and it fertilized the chemoautotrophic bacteria. So these things are ephemeral. They can be ephemeral. And so the animals are adapted not only to the weird chemistry and the extreme chemistries, but also to a habitat that goes away, that disappears, that has very high disturbance rate. So we think of the communities as resilient. This is showing an outline of one eruption in the same place. There was one in 1991 and then in 2010, or maybe 2009. And this shows the kind of the aerial extent of an eruption. That's, I can't read that. I think it's like 10, I can't tell you. 1 or 2 kilometers? I think it's 2 kilometers there. That would make sense, yeah. So we can start to learn about the geological events, what a unit magma eruption looks like on the seafloor. Pretty amazing. And we've just gone on from there. We've learned so much. Well, what happens to the biology? So we have this eruption. This was the 1991 eruption. Everything's smoking. We went back in 1992 and started laying out a grid. This is Rich Lutz, especially from Rutgers University, laying out a grid of markers. So here's a marker laid out. You see it photographed in 1993. So this is the exact same place. And these are tube worms. I know it's fuzzy, but I want to make the point that the biota comes in really fast, and it changes. So first you have tube worms, one species, then you have another species of tube worm comes in, and then mussels come in. We have a gap of data. The mussels take over the tube worms, so the tube worms are stuck on the seafloor, and then the mussels can climb on them, and they kind of overwhelm and kind of take all the good fluids away from the tube worms. 2004, it's really just mussels. So these sites last a couple of decades. It's different in different places. So on the Mid-Atlantic Ridge, the sites are much longer lived. The shrimp were there and when they were first discovered in 1985, and you go back 2010, and the shrimp are still there. It's just this massive swarming, not the same shrimp, but shrimp in the same places. So how do these, if you're going to be destroyed every 10 years, how do you maintain a population? And the way invertebrates do it in shallow water and in these deep sea sites is to have larval development. So these are examples. Here's a little crab, Megalopa, larval staged right after it's getting ready to settle on the seafloor, little polychaete larva. These are vent larvae. This is larvae of limpets, obviously a gastropod. Here's a little scallop shell. So these larvae are kicked out into the water column and they disperse. Well, that's got to be the easy part because you're in the open ocean, right? The harder part is how do you get back? And how do you get back in such large numbers? That is still a puzzle we don't know. We study gene flow, and you're going to hear a little bit about gene flow in a second. And so we know that there's dispersal, long distance dispersal over time, and these kind of stepping stone ways of moving genes around so the genetic diversity can be really high. But that's not the same as maintaining the really high densities of animals in one place. And so that's still a puzzle we haven't tackled yet, and hope to do so soon. We're starting to get the tools to be able to do that well. This is a little bit of a summary to just emphasizing that, in fact, in all the deep sea kinds of environments we think of, here's active hydrothermal vents and a slow spreading center, fast spreading center, so much more dynamic system here. So frequency and scale of disturbance on the x-axis. So these are really highly disturbed. They're also really productive. They have rapid growth rates, and they have a very high reproductive output. The contrast is thinking about the abyssal plain, just out in the middle of the ocean where it's just mud. Things go really slowly there. There's not a lot of food. It's not food rich at all. It's cold. It's just constant, relatively constant. These manganese nodules that are of interest to mining interests are in this kind of situation where they're not disturbed at all. They grow very slowly. So you think about if you're going to take something from the seafloor, well, maybe this is the place where you could get away with it without disturbing too much. But these are patchy environments. This is really very extensive environment. So there's all kinds of trade-offs to think about. And those of us who are in this business now are really, it's a real intellectual challenge and fun to think about how to manage the seafloor for mineral resources. Then there's other things, kind of intermediate, cold water corals. Cold seeps are analogs to hydrothermal vents where fluid is flowing up out of continental margins. and you get chemosynthetic faunas there. Same with whale falls. You have things that come in and use the energy from microbial production on the whales. For decades, these hydrothermal vent systems have been the provenance of where scientists work. We were the principal stakeholders. Other stakeholders were documentary filmmakers, perhaps, but we were really the only players down there. And this is covering all kinds of scientists, geologists, chemists, biologists. This was a paper we did in 2011, just kind of thinking about what the value of hydrothermal vents were to scientists. 700 cruises, and if you figure a cruise is, say an average cruise is a month long at $60,000, $70,000, $80,000 a day just for the ship, never mind the scientists and the scientific research that goes on in the laboratories at the end. It's been really a very large economic, well, from my perspective anyway, a really large industry, I guess, on the seafloor. And you can see we have hotspots. These circles are where the most cruises are. So Northeast Pacific, that's obviously US, Canada, do a lot of work there. East Pacific Rise was where we discovered vents in the first place, so we went back often, and then there was the volcanic eruption, so everybody went back there. There's now a lot of work in the Western Pacific because there's some gold-rich sites there, very interesting biogeography, and off Japan, the Japanese are diving there a lot. Not so much in the Indian Ocean, but it's starting to pick up there. So we have our favorite spots. And so for someone like me, so where do I want to go? I want to go up into the Arctic, where we don't do so much stuff, getting down to some of these sites in the Pacific Antarctic Ridge where it's, well, wherever that ridge is, where it's really nasty weather. It'd be nice to send autonomous vehicles down there to explore because, I can go, give me money, and I can take you tomorrow where we're going to find 30, 40, 50, 60, 70, 80 new species, maybe species belonging to groups we've never seen before. So there's still major, major discoveries to be made on the seafloor. So at the same time, in the ocean depths, there are mines of zinc, iron, silver, and gold, which would be quite easy to exploit. 20,000 leagues under the sea, Jules Verne, in 1870. So he got it right. There are. I don't know about the quite easy to exploit part, but he got it right that there's a lot of metal down there. So I'm going to show you, I'm not sure, let's see, I guess I want to show you, I want to get you thinking about how we think about mining, who's in control, right? So if you're in national waters, you're under national jurisdiction. But what about in international waters? Who's paying attention to that? And especially since that's where all our mid-ocean ridge is. Most of the mid-ocean ridge is in international waters. So there is an authority, the International Seabed Authority, that writes the regulations, has jurisdiction over the seabed mineral resources in this area beyond national jurisdiction. They call it the area. I like that. And so here you see examples of, in just recent years, they were able to put out calls for leases in international waters for hydrothermal vents, seafloor massive sulfide systems. China, Russia, India have leases in the Indian Ocean, and here's on the Mid-Atlantic Ridge. So these leases for exploration are being let, and they have, I don't remember the rules, 15 years to study them and then relinquish some and begin developing their mining plan for others and their environmental plans. So it's something that is of interest to both national entities and this International Seabed Authority. So what are we talking about? Why would we go after these bits and pieces of junk on the seafloor, metal on the seafloor? This is from Nautilus Minerals, which is the first mover in deep sea mining, first company to get an exploration lease. Working in national waters. So at first they began in Papua New Guinea, they've spread elsewhere. But this is data from Papua New Guinea and then a comparison Fiji Basin. So we've got land-based ores here, gold, zinc, copper. Let's look at copper. This is less than 2% copper in the land-based mines now. Historically it was higher. Manus Basin is in Papua New Guinea. Saguaro 1 is their first prospect that they would like to mine, 11% copper on average in their cores, in their test cores. Saguaro 8 is another prospect, even higher copper percent. And then they get this lovely bycatch of gold at the same time, and also a lot of zinc, and those are really interesting commodities. Fiji Basin, another area with hydrothermal vents, and again, very high quality ore. So there really is good quality ore there. It just happens to be 1,500 meters underwater. Well, 1,500 meters doesn't stop the oil and gas industry anymore, and I think it's not going to stop the mining industry. Nautilus Minerals has a lot of leases in Papua New Guinea. So Papua New Guinea, it's the country just north of Australia. And this is Manus Basin right here. So you can see Nautilus has most of the tenements in this area. I think this is old, this is from 2010, so they're going to start relinquishing some of these. The Saguaro 1 site, prospect is right over here, very close to land. They're very clever where they chose this first site. It's calm weather most of the year. Typhoons pass by it. They don't get hit by typhoons. Rabaul is a deep water port close by. Singapore, where a lot of metal, and Southeast Asia, where a lot of metal processing is done, is close by. So that's where they're working. And they have a plan for how they're going to take this ore out. Basically, you see down here, this is what, without the animals, they took out the animals in the picture, but this is basically open cut mining. And they mine with mining tools, and they show two versions here. And this is a man right here. So they're quite large, and they're very complicated machines. Having worked with watching scientists build Alvin and see how complicated it is to get it certified to dive, this is going to be quite a thing to see this go down on the seafloor and actually work reliably for a while. It'll take years I would imagine, to get it to work, to be operationally really robust. But we'll see. I mean, it could happen. And then they grind up all this stuff and they take it over to little pods, and then they vacuum it up and take it through a riser pipe up to the surface. Again, this is 1,500 meters, to a processing ship, so the surface ship. And then they do something called dewater. So basically, they have large centrifuges and they take all the water out. The water goes back down. In this case, they're bringing it back down to the seafloor. So it's well below where the tuna are in the area, and other issues of concern outside of the area of the surface that the local people are really concerned about. And on the seafloor, they just let it out down there. And it's so well filtered, they're going to be dumping less than 10 microns. That's their specification. Nothing should be dumped unless it's smaller than 10 microns. And it's what came from the seafloor goes back to the seafloor. So we think about environmental impacts of deep sea mining. So working with this company, what could be happening? So there's the physical, chemical impacts. They're going to have loss of habitat. They're going to just scrape the surfaces, so the animal habitat is going to go away. There's going to be also degradation of habitat quality, perhaps by the sediment particles raining down in the environment is one example. The fluid flux regimes, the black smoker fluid comes out from the seafloor. That will still flow out. They're not going to be able to stop that. They don't want to stop that. But it could be altered. You take away that cap of metals, and you reduce the reaction chamber. You modify the reaction chamber. So there's probably some fluid flux regime changes, and it might shift to different locations. Sediment plume and sedimentation from a little bit of overburden, light and noise on the seafloor, the bottom water nearby, and they're going to take up a lot of water and filter that. So if there's larvae in the water, they'll lose that. And then there's this little bit of, well, we don't know, but presumably a little bit of plume from the return water. So what does that mean? This is our job as biologists to think about how that could impact the biology. So first of all, when you scrape them, they're just going to eliminate and reduce the local populations. You're going to have decreased reproductive output, probably loss of larvae and zooplankton in the riser system. We don't know to what scale, how critical that is, but something we need to understand. There's a lot of rare species in these systems. So when we sample, we see the really dominant things are visually obvious. And then there's some things that are really abundant. And then there's some things when we can sample the heck out of a place and only get one individual. There's many species like that we consider rare. So we might lose some of those, whether that's good or bad. Well, whether it's bad, it's hard to know. We won't have seafloor primary productivity. So these bacteria primary producers, we would alter that. It would be decreased. It wouldn't go away, but it would be decreased, presumably. Decreased diversity, because we've gotten rid of all those animals. There might be toxic effects, altered behaviors, and then we're also interested in the cumulative effects. So I'm not saying all these are going to happen, but they're potential things. It's what we think about when we work with a company to think about how to mitigate those impacts. So again, mentioning this partnership, Nautilus Minerals came to me in 2005, back when I didn't believe mining was ever going to happen in my lifetime, mining on the seafloor. And they asked me to join them and do some, I think they really wanted me to be a consultant and consulting firm, do consulting firm work for them. I said, no, I wouldn't do that, but I would do research. And so I had research contracts with them, so you should know that I've been paid. I have not taken a salary from Nautilus, but I've taken money to do research, and I've paid students with their money. So it's been a really productive partnership, and it's an example of where, if you work together, you can get much more science out of than I could get through my National Science Foundation. It's just the opportunity was open. And the first thing, well, okay, not the first thing. Let me show you. So we're working, here's the Saguaro 1 site. This is what it looks like in cross-section. So they've drilled down to see, test drills, to see what the quality of the ore is. It's really quite small. It's not a large patch on the seafloor. So, you know, hydrothermal vents, I didn't say this, but a hydrothermal vent could be the size of this field, could be the size of this room, and you take the submarine and you know, one hop and you're over it, or the size of a football field, or maybe a couple of football fields, but they're just not really very big. And then there might be one 10 meters away, a couple of 10 kilometers away, a couple kilometers away, or maybe 100 kilometers away. So little patchy environments. So we're working here in Manus Basin. So Saguaro 1, Saguaro 2, and let me say it straight. Saguaro 1, South Sioux, and Saguaro 8 are our main study sites. We went out with the Nautilus Minerals ship and collecting tools. We were also interested in some work in the animals that live in Fiji and Lao Basin. They're often the same species, so we could make comparisons across. Here's the topography of Saguaro 1 and South Sioux. South Sioux, Nautilus Minerals has set aside as a reserve. They will not mine it. So they have a mining lease from Papua New Guinea. They have the go-ahead to do it. They just have to get the money and vehicles in place to do it. And this is a reserve, so they will not touch it, but they needed to know what it looked like. How good a reserve is it? So they asked us to compare South Sioux to Saguaro 1 in terms of the community composition. So that's part of what we did. But my first interest was in whether the inactive sites, so we have this active area, there's snails all up here. You'll see a little bit more of this in a couple of other pictures. And then all these other pictures are inactive sites where there's no more fluid flow, it's just cold. And so the animals that I'm most interested in that are adapted to the weird chemistry are gone. But then there's these longer lived things that are more general, found much more broadly in the deep sea, but they're living close by these vents. And I was interested in whether these guys ate chemosynthetic bacteria or not, right? So do they rely on the hydrothermal vents? And I was pretty certain they would not. And the way we tested this was with some stable isotope techniques. And this is just to prove I had data to support, you don't need to look at that, but to support the claim that, in fact, the tags at the inactive vents, when we looked at them, they had isotopic signatures, so biomarker signatures, biosignatures of having eaten food derived from chemoautotrophic production. So in fact, they are eating chemical, bacterially derived material primary production. What we don't know if it's coming from bacteria eating the old sulfide or coming infected from the nearby active vents, which is a really interesting question to answer. But that was my research question. I don't think Nautilus really cared too much about that, but they let me do it, and it was a surprising outcome. But what they really wanted to know was, are the same species there? So we looked at comparing South Sioux the way ecologists do. We did species effort curves, so you keep collecting more and more samples to see if you get more species, and you compare these curves, so cumulative samples, cumulative species. And we see, if we kept collecting, we haven't reached an asymptote, so we haven't really found all the species that occur there. And then South Sioux and Saguaro 1, that's the two different colors, their lines are right on top of each other. So for these inactive sulfide communities, it was a good match. If you're going to have a reserve, at least you know that at South Sioux, you're preserving the same thing that you're going to destroy in the other site. So that was good. We weren't trying to make Nautilus Minerals happy. We were just trying to discover what it was really like down there. And then we did a much more, because it was easier to sample, you have more animals, you can do a more quantitative study. So these are called hairy snails, Alvin acanca. So this patch, we would sample from patches of these. We'd sample patches of these black mussels, and then these are barnacles. We'd break off pieces of the chimney and sample those and look at the animals associated with them. And What's interesting is Nautilus Minerals allowed us to do this kind of benthic sampling in far more intense way than I've ever been supported by the National Science Foundation. And it's because they had time that they could give us. It just takes time to sample this. And so we did 3 different mounds within the Saguaro 1 site. We did the 3 different habitats, and then we did replicate samples from each of those. So we had really very intense nested studies, so we could tell whether patch by patch was different, or mound by mound, or site by site. You know, we could address all of that. And the happy answer for us was that it was pretty easy conclusion. This is looking at its kind of collapsing a lot of information about species presence and their abundance. And so here's Alvin habitat, and the white circles are South Sioux, and the black circles are Saguaro 1, and they're all mixed up. And so we know that in this Alvin habitat, they're really very similar. It's kind of the closer the circles are, that's the more similar the sites are. The same with Ephemeria, the same with Eochionolasmus. So South Sioux, in fact, is a good reserve site. It's representative of the area and serves as a nice reserve. The other thing that Nautilus wanted was South Sioux to be distributing propagules. So you have this broodstock, basically, that would distribute propagules downstream. They chose it because it's upstream. And then the question was, could it really deliver downstream? So we tackled that with genetics. We call it population structure connectivity, looking at gene flow. And again, here's those sites. And then we also compared what we saw here in Manus Basin to Fiji Basin and Laos, 2,000 kilometers separating, and then another 1,100 kilometers, so quite far apart. And we looked at a number of species. So we used multiple species and multiple genetic markers. And we chose our species, basically, they were the numerically dominant taxa, because we could sample them easily. And so there's a bias right there in the analysis, but you have to have the animals in order to do the analyses. So here's that black snail. It has a little limpet on it called Olga solaris, which is also abundant, really very abundant. So that's that limpet. We had a shrimp, Coracaris, which you see right here, a squat lobster, Munidopsis here is a squat lobster, this barnacle, Eochionolasmus. Don't you want to just say that, Eochionolasmus? Just trips off the tongue. And Volcanolepis is this stalked barnacle. So we looked at all those. The one that has the endosymbionts, that has a chemoautotrophic endosymbionts in its gill, is this black snail. So it lives right where the fluid is coming out. The rest of them are we have 2 that we think are endemic, others that are more generally found in the deep sea. They're different. Some are sessile, some are mobile, some are attached, so different styles of living. And this is students and postdocs who have worked on this. So we're looking at different markers, genetic markers, and initially, you know, kind of the very simple baseline, just to make sure we're working every individual is the same species, we do the CO1 gene, cytochrome oxidase 1. That's just a standard for species identification. And then we also did some microsatellite work and some RAD-tag work. These are just different tools to put little genetic markers on things and look at their similarity. It's not really a marker, it's looking at similarity and differences in the genetic sequences. And the microsats give you higher resolution, more data, there's more information. The RAD-tag gives you still an order of magnitude more information. We're just going into better and better genetic tools, and because they've gotten cheaper and cheaper over time. The color coding shows you where there's similarities. So if we take Ephemeria, for example, when we look at CO1 and with the microsatellite data, in Manus Basin, everything was similar. And we couldn't separate the different populations out, whether we were looking across patches. Yeah, we thought maybe the patches would be little family units, but they're not. They're all mixed up. But when we look across to North Fiji and Laos, they're very different. We could separate out. We can immediately tell the Manus Basin animals from the North Fiji and Laos animals. And which is interesting because these are separated by 1,100 kilometers, this by 2,000. No difference here, but we did see a difference there. So it's got to do with hydrography and where larvae can go and tectonic history. So for the limpet, this was interesting in that the CO1 gave us one signal for Manus and comparing with Laos, and then the RAD-tag showed us that we could distinguish Laos Basin animals. So this is comparing within a species. And maybe the most interesting for us and kind of problematic is the squat lobster, where even within Manus Basin we saw a difference. What we're starting to see now as we develop these techniques and get more data, we can start to look at directionality of gene flow, and we're starting to see a hint that, and this is by no means definitive, but we're just curious about it, that maybe South Sioux is a sink rather than a source. It's a hypothesis we still need to test. We need independent measures, but it's surprising to us, and so surprising things need extra support. So that's what we try to do. You know, Nautilus Minerals is trying to do the best they can to find the right reserve. They had all the good logic, but they're willing to listen to us. If we say, well, actually, that's maybe not the best reserve, I think they would go and choose a different site or work with us on that. So it's really a good exchange of information. And just a little bit more detail to show you what these haplotype maps look like. So here's this black snail. And we can just clearly see that all these are all different haplotypes, and the size of the circle means how many fit that haplotype. So it's really like this guy has, think of it as a wrinkled pea, and this was a smooth pea. So we can see these kind of phenotypes, if you will. I know a geneticist. I'm not a geneticist. My students are, but I'm not. I like the questions, and they help me get the answers. But we can see there's no connection. These are color-coded in shades of gray, but these two gray boxes are all found only in Lao and Fiji Basin mixed, white and gray. And these dark guys are all in Manus Basin, with one exception. There's one little guy right there. And then we have another technique to use with the microsatellites that shows us all in Manus are mixed and North Fiji and Lao Basin are mixed, but not across. So we're really starting to see natural population structure. So nobody said this, but you could think, well, okay, we're going to, if we took everything away from Manus Basin, well, that's okay because these guys are still making it back and forth, but you can't rely on that. They're not a source to feed Manus Basin. Nobody was saying they would do that. That wasn't anybody's logic, but you could see how that would work. So we're trying to parse out what this natural population structure is. Okay, this is just a transition slide. I want to talk a little bit about mitigation of this kind of mining impact and a little bit about how the larger community of deep sea scientists is thinking about this. There's in mitigation, I don't know if it's mitigation theory, but in practice, I guess, there's this idea of a mitigation hierarchy. You're going to have some kind of biodiversity impact if you do a human activity. And what you want to do is avoid impact as much as possible, but you're still going to have some residual impact. And so there you want to try to minimize that impact by maybe some clever engineering. And what you can't fix that way, then after you've done your activity, you can try to rehabilitate. And then after you've tried rehabilitating, there's still going to be some residual impact. And the idea is then you still want to end up as an industry with a net positive impact by doing some kind of offset. So this is kind of the thinking in which these environmental managers work. And so we do have some avoidance activities already going on. Part of it is through these World Hydrothermal Event Marine Protected Areas. Canada has one for hot springs off of the northwest of Canada. Mexico has one in Guaymas Basin in the Gulf of California. Portugal off the Azores. Some of these are mixed use. Portugal may even allow some mining in this area, but it's really got a lot of legislative control. United States, we have the Mariana Trench National Monument, believe it or not, with hot springs protected. New Zealand has benthic protection areas, and internationally we have the Antarctic vents that are protected. So we have some that are just set. They're not strategically designed in the sense that we are protecting different regions for different reasons, but it's a pretty good mix of things. But the community has also thought about if you're going into a region, and like, say, Manus Basin, then you want to set up, Manus Basin is pretty much its own biogeographic province. We see that because the animals there are genetically different from the ones in Fiji and Lao. And so we want to manage within that bioregion, and we really want to network. You don't want to rely on just leaving one site there. You want to have a couple of sites that are cross-fertilizing, right, where you know you have connectivity. And then ideally you'd have replicates of those networks, which is asking a lot, but that would be the ideal situation. And you'd want to include these ecologically and biologically significant areas. And also, if there's science areas that have been long-term scientific sites, you want to keep those in as well. And then from there, you define what the human uses and levels of protection are. You really have to test. Is it working? You have to keep monitoring and testing and you need to adjust. And working with industry, it's like, well, let's set aside places, but then if we find we have more than we need, can we open them up? We want to work with industry so that the best quality ore they can get, but still leave best quality natural habitats as well. So it's an interesting, thinking about the trade-offs and how to do it well. The other thing is thinking about temporal management. If you extract minerals in 2015 at a hydrothermal vent, we've seen examples where in a natural disturbance, catastrophic disturbance of a volcanic overrun, that they come back in a decade. Well, so maybe you can mine in a region one place and then just let it recover and then go mine the next site. You can't, it'll recover biologically. They're fossil deposits, so it won't recover the deposit, but it will recover biologically. Go back in 2025 and do the next site. Industry may not be able to support that, but thinking about what kind of temporal management we can do might help us out a little bit. And then rehabilitation. This is just something that I find very fascinating. So this is a document that Dr. Verlan has been, I guess you've led this, right? It's the Code for Environmental Management of Marine Mining, developed by the International Marine Minerals Society. And it includes words about rehabilitation and decommissioning plans. And so the industry itself is thinking about these things and being very progressive, that the industry should include rehabilitation in the concept design, that there should be plans and targets, that there should be monitoring, that there should be accounting for rehabilitation costs as part of the project and the costing, using adaptive management strategies and accounting for how well the rehabilitation is working. So think about it. How do you restore the deep sea? How do you rehabilitate the deep sea? Really mind-boggling kind of thing. And it was something that to the deep sea biology community, this is our world, none of us have ever thought about how to do that until very recently. And even then we're like scratching our heads, oh my gosh, how do we help industry figure out how to do this? We don't know. So we're thinking about it. Restoration happens in coastal areas. This is US examples. We have a great one, a big Florida Everglades restoration. Look at the amount of money we're willing to put into this. I can't see, but it's a lot, $13 billion, right? So, it's just remarkable how much money is going to go into restoring the Everglades. They are beautiful and valuable. Restoration goals, they're not the same for every kind of discipline. I got challenged the other night about talking about restoration because it means different things to different people. Our group has decided to call it restoration, our group being a bunch of people who met to discuss it. Conservation biologists think about endangered species and communities. Resource extraction thinks about rehabilitation. So in the US, mountaintop mining will restore the trees. Flat mountaintops are actually good in West Virginia because there's not a lot of flat land for golf courses. And so that's a positive. The government takes it as a positive rehabilitation, believe it or not. Wetland management is really functional replacement. Can you get the wetlands back and working? Landscape ecologists think about restoring ecosystem services. It's not to set the clock back to what it was, but to try to bring it back to life in some way. Increase the value of the environment after disturbing it. Setting restoration goals is a value-based activity. It's a social activity. And here's a problem for us. I don't know why I don't care about the bottom of the ocean, but I don't. And this is something we work hard to try to educate the public. The Nautilus Live, those watching a live video of the seafloor is one way. I take artists out to sea. There are people who write poems about the deep sea. So we try to reach people in many different ways. It is a really interesting place, but it's hard to get people excited about it. They don't see it from one day to the next or think about it. So we thought about decision making, and just this is an intellectual exercise of where's restoration favored, what kind of conditions. We thought about socioeconomic decision parameters, so that's this top line, things like governance benefits, ecosystem benefits. I can't even read, I don't have my glasses on. Ecosystem benefits, governance, cost, societal pressure, does anybody really care? So salt marshes, people want the fish back or they want the view shed, financial incentives through government, other wider socioeconomic impacts. Ecological decision parameters, the vulnerability, the benefit, natural recovery. If it's going to recover naturally, why would you invest in restoration? Large relative ecological impact. And then technological, is it going to succeed? If you spend a lot of money, will you be successful? Is it feasible technologically? Or maybe it's just you want to advance the technology. So we looked at salt marshes and deep sea coral systems. This is thinking of the Darwin Mounds just off the British Isles, and hydrothermal vents. And so this whole nice green bar here makes it pretty reasonable to go after a salt marsh and restore it. Everybody likes it. Lots of people see it. Societal value. You can do it ecologically, natural recovery. You know, when you put up a bulkhead, it's hard for a salt marsh to come in, so you have to tear down the bulkhead and put the salt marsh back. And it's feasible. Deep sea corals, much more pink. You know, there's no governance that says you have to do it. Financial incentives, none. No wider socioeconomic impacts to restore deep sea coral. But there's some societal pressure. They're really very vulnerable. They're not going to recover naturally, probably, for a very long time. Those are old growth forests, basically. And we probably could do it, but it would be very costly. And then hydrothermal vents were kind of down here in the red zone. There's, you know, it's the benefits, technological advancement for sure. Is it feasible? Probably, but it's going to cost some money. Success, we could probably we could be successful in some goal, as long as it was a limited goal. So anyway, it's kind of an interesting thing to think about. Why would you do it? And yet, the mining industry is thinking about doing it. In part, technological advancement, in part, corporate responsibility. So there are societal pressures to do this, to think about this kind of thing. So how would you do it? I'm going to give you an example of what we're thinking of doing in Saguaro 1 with Nautilus Minerals, what Nautilus Minerals is planning to do. I'm presenting it as a 5-year academic program because that's the world I know and the accounting I know. Nautilus Minerals would give you a different number and probably a much more favorable number. But this is the immediate objective would be to establish some mounds and the animals, put the animals back. So here's the animals we want to have back, the active sites with snails and the inactive sites with corals and things. And so you want to do 2 sites, active, inactive, 4 conditions, high, medium, low density, transplants. You're moving from in front of the mining tool to behind the mining tool to where you're trying to recover things. And success, you know, pretty modest. You get survival, growth, recruitment, and increased associated diversity. So that's modest. You're not getting the whole system functioning, but you're getting some good positive benefits. And here's the artist concept of this. They have already placed on the seafloor some of these rock-filled pyramids to stabilize a 3-dimensional structure. And the idea is these little ROVs, while they're not doing other activities, could go off in front of the vehicles, collect snails, put them back over on this site, kind of a little bit of rescue transplant, but also just to learn how to respond and how to, this little tiny baby step towards restoration. It's really engaging. It's caused it's created a lot of controversy in the scientific community and the social community about investing time and money in this kind of activity over other things that you could do, and also just, you know, the experimental protocol. You know, how do you design it to be a really good experiment and meaningful? And so again, I emphasize this is an academic restoration project where, like, I'm the project manager. I get a month a year, say for 5 years. That's pretty luxurious. Lab tech for a full year for 5 years. Maybe he or she is not always working on this project, but you got to write it up, and that's the person who does a lot of the data analysis. You have to build the 3D substrata, so that's got a cost. You've got some supplies, time-lapse cameras to monitor what happens. You have to have cruises. Here's where the money comes in. This is the expensive stuff. And so total, you know, $5 million over 5 years for if you were to want to fund me and my lab group to go do this experiment on the seafloor. Oh, interesting. I have another slide that compares this cost on a per hectare basis to salt marshes. And it's, you know, it can be 3 to 4 orders of magnitude more expensive than restoration on a per hectare basis. Part of that's because this little study site is so small. The hectare, you know, you're only doing a fraction of a hectare. But there's also efficiency. So engineers and scientists can work together to build vehicles that are specialized just to do restoration techniques, right? So it doesn't have to be the scientific research, you know, all the bells and whistles. It can just be down there to just routinely go after a single task or set of tasks. interesting concept, thinking about restoring the deep sea. There's other places much more difficult to restore. If you think about those down in that lower left-hand corner of the resilience graph I showed with those manganese nodules, the nodules themselves form very, very slowly. The animals that colonize them are slow growth, old growth animals for the most part. And it's a very, the mine area is really large. How do you restore that? Or how do you rehabilitate that? So it's, maybe you don't. Maybe some places you do, like a hydrothermal vent that somebody really cares about, or just for experimental purposes. Okay, so this is kind of coming to a close. There is a group called DOSI, Deep Ocean Stewardship Initiative, that's thinking about how to do this union of experts and ideas. We're really trying to work with industry, so industry's involved, as well as academics, as well as civil society, NGOs, non-governmental organizations, thinking about how to do environmental management, environmental integrity, information sharing, and then a suite of different working groups to work with the International Seabed Authority, to work with education, outreach, capacity building, different kinds of efforts. This is really, in 2005, I felt like I was the only person who was working in this area, and I got nasty mail for working with a mining company. But I think the larger community now is getting involved. I know it is, and including, I was just at a meeting 2 days ago of what's a project called IMarEST that is thinking about exactly the same stuff, environmental management of mining in the deep sea, thinking about the scientific knowledge gaps that need to be filled, thinking about the legal issues, policy, how to use the science, social science and natural science to inform policymaking. And that's an EU program. And so I think it's going to be really the leader, it's going to provide the leadership in this world. So it's going to be in the news, I think, and you'll hear a lot about what we're all up to. My final words: in the deep sea, there do live strange creatures of extraordinary beauty. They're exquisitely adapted to extreme environments and seabed, and at the same time, there are seabed resources that are important to society. We may choose to disturb these systems in the interest of society, for our cell phones, for the things we need and want, to gain economic advantage or also strategic advantage for some things that nations don't have on their own land. But I think we have to make these choices with attention to mitigating the impacts and maintaining the ecosystems. So we can't just stick our heads in the sand and ignore the problem. We have to be part of the solution as scientists. So that's it. Thank you very much for your attention. And